The COVID-19 pandemic has put an abrupt stop to many daily activities over the past year. However, research at the Wood Hudson Cancer Research Laboratory has not been phased. Focused on education just as much as on research itself, the laboratory offers the Undergraduate Research and Education Program, or UREP, in which students have the opportunity to gain first-hand experience in the lab as well as presenting their findings to other researchers. In fact, 3 undergraduates this year, Anna Oliverio, Angel Rapp, and Brianna Hoegermeyer, were able to present their findings at the 2021 AACR Virtual Annual Meeting. I had the opportunity to talk to them.
So my name is Anna Oliverio. I am now officially alumni of Thomas More University with a biology major with a concentration in cellular and molecular biology and a minor in chemistry.
Hi, I'm Brianna Hoegermeyer. I'm a rising senior at Thomas More University with a degree in biology as a Bachelor of Science, and I'm also going for a concentration in cellular and molecular biology.
All right, awesome. So what was your research project about?
So our research project was it's a new perspective on research. It's not genetics, but it's the metabolome. So it's how the cell's able to regulate its metabolic processes inside itself. And as researchers become more familiar with the metabolome inside cancers versus normal cells, they realize that some cancer cells are able to shift certain metabolic pathways. So we were specifically looking at breast cancer and how it heavily relies on the serine biosynthesis pathway inside the cancer cells. And then I'll let Brianna kind of tag on there and finish off the rest if she wants to add anything.
Sure. So indeed, we were looking at the serine biosynthesis pathway, and we were specifically looking at Phosphoserine Phosphatase, which is the third enzyme in the pathway, which cleaves a phosphate group off of the precursor and gives you the serine. So what we were curious was if there was a different expression of serine Phosphoserine Phosphatase in cancer cells from breast cancer versus non-malignant cancer types or normal breast tissue. So Anna had done a lot of background research and saw that it was pretty common for the serine biosynthesis pathway to be upregulated in certain types of cancer. So we kind of went off of that, and what we did is we used paraffin-fixed formalin-embedded tissues, and from patient samples from the sample repository at Wood Hudson, and we actually rehydrated those, did antigen retrieval, and stained specifically with an antibody for the phosphoserine phosphatase and looked to see if the expression was different.
Their study found that phosphoserine phosphatase is strongly associated with metastasis in breast cancer, which indicates that the tumors upregulate the enzyme in order to proliferate. This is also consistent with a lot of studies done on serine and cancer. So in cancer research, a lot of the time you hear serine, like this specific amino acid, being thrown around a lot. So what significance does serine have, especially to your project?
Serine is important for many things. It's incredible what a cell can do with just one amino acid, besides building the proteins. So serine can be shifted into other metabolic pathways that synthesize things such as the nucleic acid components. It can be shifted into pathways. There's one of them called the carbon pool. It's like a cycle of carbon, so the cell can like shift carbon where it needs it to go. It's even used to make sphingolipids. So it's like all these different components within the cell. So it's very convenient and helpful for a cell that wants to keep replicating to be able to have a source to keep building those materials and dividing.
Their findings aren't limited just to academic purposes. There are real-world applications, especially to targeted therapies. So what do you think the most significant part of your project was? So what do you think it means for the future of cancer research?
That's a really good question. I would say the most important thing that stuck out to me is finding unique and creative ways to treat cancer and end the use of chemotherapy, where we're basically just poisoning. it makes the patient so sick. It attacks the good as well as the healthy cells. And to find treatments that are more efficient at targeting specific cancer characteristics, such as their unique metabolic properties that are different from a healthy cell, it offers a new opportunity to treat cancer in a safer and more specific and efficient way. And I think it'll make cancer treatments cheaper. It will increase survival rates. I'm probably being a little bit optimistic, but I think research in this respect, where you're looking at the metabolome in general, it'll take a long time probably to build it up to that point. But I think it's worth seeking and looking into for those reasons.
All right, thank you very much for your time. And I'm going to stop recording now, but I appreciate taking the time out of your day to talk to me. Treatments involving the metabolome are still in their infancy, but progress in its research is indicative of a much greater change in the medical community. Thanks to researchers like those at Wood-Hudson Cancer Research Laboratory, we're now a few steps closer to victory in the fight against cancer. Thank you very much for watching. Angel Rapp wasn't available for interview for this video, but her work was still integral to the project that they did. So be sure to check out Wood-Hudson Cancer Research Laboratory's website to read more about these researchers and the project that they did.